This is about the Kelvin cycle. Woo, Kelvin cycle. And named after scientists that studied much of these steps out in Berkeley, California in the 1940s and 50s. It used to be called the dark reaction, so that's something you may have even still learned in school. And the idea is we have light reactions, we have dark reactions. The light reactions require the light, take place in the light. The dark reactions could take place in the dark. Normally they're taking place at the same time as the light reactions because the light reactions are producing ATP and they're producing NADPH. And the ATP and the NADPH are used to drive chemical reactions in this pathway. And again, we don't need to memorize the names of these molecules or the enzymes that catalyze each of these steps. But we should go back to our Chapter 8 knowledge of energetics. And what we could say is that what's going on here is this is probably an exergonic reaction, energy releasing spontaneous reaction. And what it's doing is it's helping set up a reaction that requires energy. So we're using steps that are steps in chemical synthesis that are exergonic or energy releasing in order to set up the precursor molecule substrate that is going to be pushed up a endergonic reaction, and we're going to couple that endergonic reaction with an even more exergonic reaction. So what can we say about this enzyme that catalyzes this reaction? Again, we don't need to know the enzyme's name, but it's going to grab hold of the substrate in its active site. It's going to catalyze the synthesis of a product, and in doing that, this enzyme is going to also bind ATP, and it's going to use that energy of ATP released to drive this chemical reaction forward. And so we could add up the net delta G of this reaction, and the net delta G will still be negative, so that this is an overall net negative spontaneous reaction. It's like the ATP is water flowing downhill, and we can use it to pump a little bit less milk uphill. So we have a big negative delta G that can be used to drive forward a small positive delta G. And the positive delta G, the pumping milk uphill, is the synthesis of the sugar. So we're synthesizing sugar. It's actually a 3-carbon sugar that's the product of the Kelvin cycle. So this is called a C3 sugar. And it's going to go on to be used to synthesize glucose and other molecules. And but this enzyme can actually couple this chemistry, so we have an overall net negative delta G, so that each of these steps in synthesizing sugar, which contains potential energy, contains the calories that we think about, right? Because we could just turn around and eat this right now and then metabolize and release those calories from those sugars. So the calories are being put in the Kelvin cycle, and overall we have an overall increase, an endergonic reaction taking place to synthesize sugar, and it's being driven by these energy sources from the light reactions. Boom, like that. So we have this coupling of energy reactions and an overall net negative delta G. We can put numbers in there. We learned that you release about 7.3 kilocalories per mole when you hydrolyze ATP, and so that's how much energy released here. So you'd say, how much energy do you think it takes to drive this substrate to this product? And the answer is, well, I think it's positive, but I think it's less than 7.3. I think it's less than 7.3, so that when we add these together, we get an overall net negative delta G, so the reaction overall is still spontaneous. So this is occurring spontaneous because energy is being input. And that's the bottom line of the Kelvin cycle. We don't need to learn the details. The details of the chemistry are actually really complex because, see, it's a cycle, so it's not just a simple linear pathway where you're like getting sugar, but rather you're adding one carbon to an intermediate as you cycle through this repeatedly and build these sugars. So the chemistry is complicated. I don't want to say complicated. The chemistry has lots of steps and stories to it. And we don't need to learn those in this course. We really should follow the energetics and the logic of the pathway in order to see this connection between synthesis of sugar, anabolism of sugar, and then eventual catabolism of sugar by either the plant or an animal that eats it. Last thing is there's one enzyme that we should learn the name of, and it's called Rubisco. And it's because Rubisco is a really famous enzyme because one of its substrates is CO2. And this is where CO2 carbon is fixed. And it's really pretty fascinating because, you know, when you see a tree, that tree is solid, and if you run into it, you are going to get hurt. But this tree's solid nature was previously mostly gaseous CO2. Trees are just really, really thick air, like that. And so are we. And because the tree has grabbed CO2 from the air and fixed it and turned it into this solid sugar form. So Rubisco is a famous enzyme, and we'll see that Rubisco is a famous enzyme in a switch story that the book talks about at the end of chapter 10. So again, you know, here's the basic of the Calvin cycle. It's happening in the stroma. Do we have that anywhere on here? So these are just floating around in the stroma of the chloroplast. And then we're going to talk about a switch. And here's the switch. The switch is that, just to go back to this, the switch is that Rubisco is pretty good at fixing CO2. However, this is happening inside leaves. And the leaf has these stoma. We saw in the very first slide, it has pores in the leaf that are open and allow carbon dioxide to come in and oxygen produced to leave. And in these, the reason that these have these pores is because these guard cells close in the heat of the day. They close to conserve water. So the plant loves to, you know, get the CO2, fix the CO2 with Rubisco, produce oxygen by the light reactions. But in having that pore open, there is evaporative water loss. And so the plant will close the pores when it's hot and a lot of water would be lost. What happens then is that in the heat, inside the cell, inside the leaf, O2 builds up and CO2 decreases. Woo. And when that happens, Rubisco, all the Rubisco on our planet, and imagine how much there is in all of the plants, right? These are all eukaryotic plants that have Rubisco. And all the Rubisco is vulnerable under these conditions to actually run in reverse. And instead of fixing CO2, it actually releases CO2, and in doing so, it uses oxygen. So under high oxygen levels and low CO2, like in the heat of the day, when the pores are closed, Rubisco runs in reverse and energy is lost. You know, and that's really tragic, but it loses, the plant's willing to lose energy because of this problem in order to conserve water, like that. And this process, this phenomenon is called photorespiration. And it's a big problem in many plants. What plants is it a problem with? Trees in Maine. So generally, big plants that are in northern climates on our planet. So plants in this region, they don't see that much heat. In this region too, around here, of course, they don't see that much heat. So there are some summer days, right, where it gets really hot, and you would think that the plants would be growing a lot, but they're not, because they're undergoing tremendous amounts of photorespiration. And these plants are referred to as C3 plants. And C3 plants are generally living in these environments. And so in Chapter 10, the focus of this switch in the photorespiration story is to show you first that there is a limitation in C3 plants, and then to show you that there are plants that evolved to live closer to the equator, more hot, dry days, hot environments, hot, dry environments, that have evolved solutions to this problem. And those plants are called C4 and CAM plants. And the book ends Chapter 10 just with a little story of their evolutionary success. C3, C4, and CAM plants all have Rubisco, and Rubisco always has a problem with photorespiration. So C4 and CAM plants have evolved a solution to photorespiration by some other means. And here are the means. It's shown in this figure. So here are the C4 plants and the CAM plants. And C4 plants, when we think of them, we could think of like grasses, and we could think of like cacti as CAM plants. There are lots of examples, but those are nice ones to think of, because you know there are some environments where grasses can grow, where it's hot and dry, but there are no big trees. And one of the big reasons is because of this problem with the C3 plants, trees, and photorespiration. Also, cacti and succulents can grow hot and dry. And how do these plants succeed? How do these types of plants avoid the problem of photorespiration? And they evolved two solutions, and it's just cute to see them. And the two solutions are that the CO2 is fixed initially by other enzymes than Rubisco. And they came up with two different solutions. In C4 plants, photorespiration doesn't occur because they evolved another layer of cells. The mesophyll cells are where the CO2 is initially fixed. And so they physically separate the low CO2, high oxygen environment on the hot, dry day inside the leaf. Mesophyll cells physically separate that from the Rubisco. So with CAM4 plants, we get a solution to the problem that involves space. You know, we're going to physically separate Rubisco from the atmospheric conditions inside the leaf on a hot, dry day. Then CAM plants evolved a different solution, and that is the solution is we're going to solve it by fixing at a different time. And so they do that at night. The CO2 is fixed initially at night and held in organic acids, so that in the hot, dry day, what happens is that CO2 would go down, O2 would go up, but Rubisco is protected because now these molecules release their CO2, and so the CO2 levels don't drop inside the leaf cell. They're maintained at a high level, and photorespiration is avoided. Boom, like that. And that's that at the end of Chapter 10, just to show you that. And it explains a lot of patterns of organisms in the ecosystem. And you remember, our course is all about stuff that happens within organisms, and the second semester is about organisms in their ecosystem. And this is just a really nice example of how molecular level things affect ecosystems. And that's the end of this one, and Chapter 10.